Welcome back, guys. Today we are going to discuss this topic in physiology of skeletal muscles. Okay. Previously we have discussed the nerve cells, the characteristic of nerve fibers and their classification and also their injury. Now today we are going to move with skeletal muscles. So we are going to start skeletal muscles with some basics. Okay. The first basic will be the arrangement of the different fibers in our skeletal muscle. Okay, so first we're going to see the fibers. Okay, this is a single fiber of skeletal muscle. The single fiber is surrounded on the outside, okay, the outside by endomysium. This is endomysium. Okay, this is our fiber. Okay, now a lot of fibers, there are a lot of fibers, not a single fiber. These are covered by a structure on their outside. Okay, now this is called our perimysium. Single fibers covered on the outside by endomysium and a lot of fibers covered on the outside by perimysium. Okay, now both of these are together covered on their outside by this single layer. Okay, now this single layer composing this covering of endomysium and perimysium, this is called our skeletal muscle. This is our skeletal muscle. Okay, now this skeletal muscle further, okay, covered on the outside. Sorry, that was not good. It's further covered on the outside. Okay, now this layer is called our sarcolemma. The sarcolemma, we must have heard about this before. The sarcolemma covers our skeletal muscle on the outside. This is the penultimate layer because there is a outer layer outside. Okay, all the sarcolemma covering the skeletal muscle, covering the perimysium and endomysium, is finally covered on the outside. Okay, covered on the outside by this final layer called epimysium. Okay, so this is the overall arrangement of different structures of inner skeletal muscle. First, there is a single muscle fiber covered by endomysium. Now, couple of muscle fibers are covered by a perimysium, and both this endo and perimysium are covered on the outside by a single layer. Now, this is composed of a skeletal muscle. After this is finished, skeletal muscle is covered on the outside by penultimate layer of sarcolemma, and the whole structure is covered on the outside by epimysium. So endo, peri, and finally epi. So this is the arrangement of fibers and layers in the skeletal muscle. Moving on, we are going to see the functional unit of skeletal muscle. Okay, functional unit of skeletal muscle, that is our sarcomere. Okay, the sarcomere is our structure, is the functional unit of our skeletal muscle. The structure sarcomere is composed of actin and myosin. Actin and myosin is the functional unit of skeletal muscle sarcomere composition. So we are going to see in details in structure of this sarcomere. Okay, so this is our structure of our sarcomere. Basically, we have to see one by one what are the components of this sarcomere. So on the extremes, these two lines, okay, these two lines on the extreme, these are our Z lines. Okay, Z lines. Then in this Z lines on the extreme, in this middle, this is our M line, M for middle, M line, M line. Okay, so we have seen these on the extreme, these are Z lines, and on the middle, M for middle, M line. Simple. Now these green structures, okay, this green filaments, these are actin filaments, these are thin filaments, actin or thin filaments. Same goes for here, actin, thin filaments. Okay, same for this, same for this. Okay, now this actin, which is a thin filament, and this Z line, they are connected together here at this angle by alpha actinin. Actin, so connected by alpha actinin. Alpha actinin joins Z line along with actinin. Z line and actin are connected by alpha actinin at this junction. Okay, moving on, there is another protein, okay, joining the Z lines to M line. Okay, from Z lines to M line. Okay, this is our titin protein. Titin protein. Okay, this titin protein, this structure, this arrangement is spring-like, so it's a spring-like protein. Okay, running from Z line to M line. Running from Z line to M line. It's a spring-like protein running from Z line to M line. Titin protein. Okay, this is alpha actinin connecting Z line to actin. Okay, now this, moving on, we have another protein here throughout. Okay, another protein here throughout. Okay, so across this. Okay, so across this is our nebulin protein. Nebulin. Okay, nebulin protein. What is the function of this nebulin protein? This nebulin acts along the actin protein. Acts along the actin protein. Okay, this nebulin, remember, regulates the length of actin filaments. This nebulin regulates the length of actin filaments. This here, this is the nebulin protein. Okay, so we have so far seen 3 proteins: alpha actinin, titin protein, and nebulin protein. Okay, now one last protein we'll discuss. Before that, we're going to see this region. Okay, this, what is this band? This band here, this band right here is our thick filament. Thick filament, or our A band. Okay, or A band. This thick filament or A band. This A band consists of, remember, our thick filament myosin along with overlapped actin filaments. Overlapped actin filaments. Thick filament A band consists of thick filament myosin along with overlapped actin filaments. These actin filaments are overlapping here. Okay, now following this, this complete band is our thick filament. which is our myosin. So we have discussed before actin and myosin is complete filament. Okay, now finally we are seeing this region from here to here. Okay. This here to here is our unoverlapped region of our thick filament. This is our overlapped region by our actin filament, but this region is unoverlapped. So this region is our H zone. H zone. Okay, H zone. This is the non-overlapping by actin of myosin. What is this H zone? This is the myosin filament which is not overlapped by actin. So this region is not overlapped by actin, and this also consists of our middle M line. So this is the H zone, non-overlapping of actin by actin of myosin. Okay, so this was our structure of our sarcomere. First we have our end Z lines. Okay, now with these end Z lines, in between, in the middle, or M for M line. So the periphery and the middle are over. Z lines are innervated by actin, which are our thin filaments. So we have two filaments, thin and thick. Actin is our thin and myosin is our thick. Z line and actin are connected by alpha actinium protein, or first protein. Okay, now Z line and M line are connected by titin protein. Remember, it's a spring-like protein running from Z line to M line. Okay, next our third protein is our nebulin protein. This runs across the actin protein, which regulates the length of actin filaments. This protein. Okay, I did it in dotted one. Okay, and then the fourth protein is the fourth protein we are going to discuss is here. Okay, fourth protein is our meromycin. Meromycin. Okay, or this is also called, instead of meromycin, we now call this as myomycin. Okay, this myomycin attaches, what is this, M line to, what is this, our thick filament myosin. So M to M, myomycin from M line to myosin. This is our fourth last protein to be discussed. Okay, following, this is our thick filament. This is our A band. A band is our composing of myosin and overlapped actin filaments. These actin filaments overlapped. Okay, and this is the finally H zone. This is our M band not overlapped by A band. Okay, so this was our entire structure of our sarcomere with their filaments. A bit complicated one, but if you practice one or two times, it will be simple for us. Okay, moving on, we have seen the sarcomere as the functional unit of skeletal muscles, composed of actin, thin filament, myosin and thick. Okay, now this sarcomere contracts. Okay, this contraction. What is this contraction? This is changes in sarcomere. Changes in sarcomere. Okay. Now, what are the changes in sarcomere occurs? Contraction, remember, first is Z lines comes closer. Z lines comes closer. Okay. I band and H band length decreases. Second point, I band and H band length decreases. Finally, our A band length remains same. This is A band. Couple comprising of thick myosin filaments along with overlapping actin filaments. This length remains the same. Most important point: A band length remains same, I band and H band length decreases, and Z length comes closer. And what is this, our sarcomere? Our sarcomere is from one Z line to the other. As in two Z lines at the extreme, one M line in the middle. So the sarcomere is consisting from one Z line to another Z line. So this is sarcomere. So contracts, changes in sarcomere. Z lines comes closer. The length of the sarcomere decreases. I band and H band length decreases, but A band length remains the same. Okay. So the features of contraction of sarcomere. Moving on, see bit about the two proteins, actin and myosin. Okay, actin and myosin. In actin, you know, this is the thin one, the thin complex. Myosin is our thick complex. Okay, this myosin has a, remember, fixed length of 1.6 micrometer. Okay, this myosin is composed of two heavy chain and four light chains. Myosin is composed of two heavy chains and four light chains. Okay, now another fact to remember here is myosin head has ATPase activity. Myosin head has ATPase activity. So there's a brief discussion about actin and myosin. Myosin particularly two heavy chains and four light chains, and head has ATPase activity. Okay. Now moving on, we'll have discussion on skeletal proteins, skeletal muscle proteins, skeletal muscle proteins, skeletal muscle proteins. So these are actually four types here. Okay. First type is our contractile proteins. Contractile proteins. Okay. Next is our support proteins. Support proteins. Third one is our regulatory proteins. Regulatory proteins. Fourth one being our relaxation proteins. Relaxation proteins. Contractile, support, regulatory, and relaxation. First we will have contraction. For this contraction we need some support proteins. These two proteins requires regulation, and finally we have relaxation, for which we have proteins. Now these contractile proteins are microfilaments. What are these microfilaments? We have already discussed before, actin and myosin. Actin and myosin. The rest of this we will discuss on further discussions. Okay, actin and myosin are the microfilaments. Okay, we have seen here both two. Okay, nothing much to discuss here. Now we'll start with our second type of protein, our support proteins. That is support proteins in our skeletal muscles. Okay, so starting with our support proteins. First is our desmin. Protein name is desmin. Okay, now this desmin is an intermediate filament. Intermediate filament. Okay, the desmin is a tumor marker for sarcoma. Sarcoma, carcinoma of skeletal muscle. Desmin is a tumor marker. Okay, it's an intermediate filament. Second support protein is a dystrophin. Dystrophin. Okay, this dystrophin forms dystrophin glycoprotein complex. Okay, dystrophin glycoprotein complex, DGC. Okay, how this works we're going to see here. Okay, we're going to draw some structures first. Okay, then we'll understand why are those for. Okay, this is another one. This is another one. Finally, this is another protein. And okay, so let's start with the discussion. Okay, first we draw two lines here. Okay, first two proteins we have drawn here. These are sarcoglycan, okay, and these are sarcospam, sarcoglycan and sarcospam. Okay, now these two proteins are drawn here. This is our beta dystroglycan, and this is our alpha dystroglycan, and they are attached here. Alpha and beta dystroglycan are attached to this alpha dystroglycan. Another protein is attached called laminin. Okay, laminin. Okay, now to inside of this, so the beta dystroglycan, this molecule, this here is our dystrophin molecule, or dystrophin protein is here, and to this dystrophin here are attached it are actin. To this dystrophin our actin is attached. Finally, these proteins, these are syntrophins. Let me write this again. Syntrophins. Syntrophins are required for attachments. So the purpose of drawing of all this is to make you understand that this is the structure of how dystrophin is attached to various proteins. There is sarcoglycan and sarcospam, to which this beta dystroglycan and alpha dystroglycan are attached. To this alpha dystroglycan, laminin protein is attached. To this dystrophin, actin protein is attached. And for all these attachments, syntrophins are required. Okay. So what is the action of this dystrophin? Dystrophin is our anchor protein. So the support protein is our anchor protein. Okay. Now this dystrophin is absent. Okay. So supportive function is lost. Support is lost, which will lead to this condition called Duchenne muscular dystrophy. Duchenne muscular dystrophy. Dystrophin Duchenne muscular dystrophy, DMD, and this is a fatal condition. This Duchenne muscular dystrophy is due to absence of dystrophin, which is anchor protein, and this is a fatal, very fatal condition. Okay, and here there is muscle weakness of skeletal muscle, muscle weakness of skeletal muscle. Okay, for example, there is diaphragm weakness, main respiratory muscle, diaphragm weakness. There is respiratory muscle paralysis, respiratory muscle paralysis, which is one of the causes for death. Diaphragm weakness, diaphragm muscle paralysis. Okay, Duchenne muscular dystrophy due to absence of dystrophin. Diaphragm muscle weakness or respiratory muscle paralysis, which will lead to death. So that's why it's a fatal condition. Okay. Now these are two of the supporting proteins. Okay. This is a desmin and then dystrophin. Desmin is an intermediate filament. It's a tumor marker for sarcoma. Okay. Following on for the third type of protein is a regulatory protein. Before that, we have another supporting protein, which is a titin. The titin protein was discussed in the sarcomere spring-like structure. Titin is also called muscle spring, which was connecting the actin to the M line, the actin filaments to the M line. This is providing classic support to muscle, classic support to the muscle. Okay, this titin is the largest protein in humans, and we know this extends from Z line to M line, from Z line to M line. This attaches myosin head to Z line. Okay, this is our Z line, this is our middle M line, here was our myosin. Okay, this was our titin protein. Okay, attached myosin head to the Z line, which is the largest protein in our humans. Okay, this titin, okay, remember, keeps actin and myosin in place, keeps actin and myosin in place, side by side arrangement of actin and myosin, by side by side arrangement of actin and myosin. It keeps the actin and myosin in place. Okay, which titin protein, important protein, asked many times before. Okay, so this was all about our three types of support proteins: desmin, dystrophin, which absence leads to Duchenne muscular dystrophy, and our titin. Okay, following on to our third type of muscle protein, regulatory proteins. Regulatory protein is an important discussion, so listen carefully. Regulatory proteins. Okay, these regulatory proteins are two. Okay, we all know them: tropomyosin, or classic tropomyosin, and other one is troponin. Tropomyosin and troponin. Okay, so how this tropomyosin is arranged, we'll see. So these are actin filaments, small actin filaments. Okay, the actin filaments have on the top this small, what are these? These are our active site. What is this active site? The active site are myosin binding site. Myosin binding site. The active site of the actin filaments is myosin binding site. Now this tropomyosin, it does what? It runs along all the active site. It basically covers all the active site of the actin. Okay, this is our tropomyosin. Okay, covers the active site. Covers the active site of actin when in resting phase. In resting phase, so that there will be no contraction. There will be no contraction. Okay, and remember, one tropomyosin covers 7 active sites. This is the function of tropomyosin. It covers the active site of the actin, preventing contraction. And what is this active site of actin? These are myosin binding sites in the resting phase. Okay, remember, in the resting phase, so no contraction. This is our tropomyosin function. Now moving on to troponin, the second regulator protein. It has 3 subunits. Okay, first is troponin T. Okay, what is this does? Binds to tropomyosin. T for T, tropomyosin, so T type. Okay, then there is troponin I for inhibitory. This is inhibitory. Okay, what is this inhibits? This inhibits actin and myosin interaction. Actin and myosin interaction. Troponin T, troponin I. Final form is troponin C. Okay, C for calcium. This subunit binds to calcium ions. This is most important component in the contraction of our skeletal muscles. So we can remember by this mnemonic ITC. This ITC company. Troponin I, inhibitory, inhibits actin myosin interaction. Troponin T for tropomyosin, binds to tropomyosin. Troponin unit T binds to tropomyosin. And finally troponin C binds to calcium. C is for calcium. Okay. Now the final type of protein we have is, after regulated proteins, relaxation proteins. For contraction and regulation, we have relaxation proteins. Okay, these relaxation proteins, we have calcium pumps called Sarca pumps. CRCA, Sarca pumps. Okay, what is this? Sarca is sarcoplasmic reticulum. Sarcoplasmic reticulum calcium ATPases. Okay, very important pump. How it's important, we will see in further phases. So this is one of the relaxation proteins, or Sarca pumps. Okay, so basically these pumps do what? This pumps actually helps in taking back the calcium inside to the sarcoplasmic reticulum after the contraction is over. After contraction, we have relaxation phase. So the Sarca pumps does what? It actually pumps the calcium back inside to the sarcoplasmic reticulum. And if this pump is non-functional, we will have various complications which we will see after our discussion. Okay. Next is, we have our contraction and relaxation of skeletal muscle. So let's begin with this. Contraction and relaxation of skeletal muscles. Okay, contraction and relaxation of skeletal muscles. So here we have, first we have an electrical event which is our action potential. The action potential is our electrical event. Okay, this is our excitation part, and then after electrical event we have our mechanical event. What is this mechanical event? Contraction of our skeletal muscle. So here contraction. Okay, now this electrical event action potential is conducted by, remember, alpha motor neuron. Now these two, okay, electrical event and mechanical event, which are excitation and contraction, are coupled together. They are bound together. Okay, they are coupled together by this ion, calcium ions, the single most important ion in contraction of skeletal muscles. So the electrical mechanical event are coupled by calcium ions. This phenomenon is simply called in general as excitation contraction coupling. Very simple, excitation contraction coupling. This is called our excitation contraction coupling. So we will see both one by one what happens, but before that we are going to see what is a motor unit. Okay, what is a motor unit? So let's assume that there is an alpha motor neuron, single alpha motor neuron. Okay, the single alpha motor neuron innervates multiple muscle fibers. First muscle fiber, second muscle fiber, third muscle fiber, going on, multiple muscle fibers. Okay, now this single alpha motor neuron, okay, combined with the muscle fibers innervated, single alpha motor neuron combined with the muscle fibers innervated, this comprises of our motor unit. What is motor unit? A single alpha motor neuron here plus the muscle fibers it innervates. Through its branches it innervates multiple muscle fibers. This is our motor unit. With this discussion of motor unit, we go to our first phase of excitation contraction coupling, that is our excitation part. The electrical event, that is our action potential. Okay, very simple. So where does this action potential or excitation event takes place? In the neuron, in the presynaptic neuron. Okay, so we'll draw this simplified diagram of a neuron. Okay, so this is cell body, this is the axon. Okay, this is the axon terminal, and this axon terminal we have our, here we have our muscle fiber. Okay, so this is our alpha motor neuron. Okay, so let us start with it. So first there is depolarization. Okay, there is depolarization, and what is this depolarization? Due to very simple sodium influx. Okay, we'll go one by one. First we'll see a diagram, then we'll write in the following steps. Okay, now this sodium influx occurs through here. Okay, here sodium influx into the neuron occurs through here. Now this is a voltage gated sodium channel. This is a voltage gated sodium channel through which sodium enters. Now positive ion enters into the system. The resting memory potential of the neuron, as we know, is minus 70 millivolt. A positive ion enters, so the negativity inside decreases to what value? Remember, minus 40 millivolt. This minus 40 millivolt. Why this 40 millivolt is important? Because at this 40 millivolt our calcium channels open. Here we have our all important calcium channels through which calcium enter. Calcium. Okay, minus 40 volt, voltage gated calcium channels enters or opens. Voltage gated calcium channels opens at minus 40 millivolt. Depolarization from minus 70 millivolt to minus 40 millivolt. So calcium enters. After the calcium entering, why this is needed? Because here we have our acetylcholine vesicles. Primary neurotransmitter, acetylcholine vesicles. This calcium causes release of acetylcholine, okay, with the process of exocytosis. The calcium causes release of acetylcholine vesicles. Okay, this acetylcholine vesicles get released and they act here. We have receptors here for acetylcholine. They act, acetylcholine act and causes contraction. Very simple basic thing here. And this is our skeletal muscle. This is a skeletal muscle. Okay, this was the total gist of it. First, there is depolarization due to sodium influx. This sodium influx occurs through voltage gated sodium plus channels. Okay, voltage gated sodium plus channels. Sodium enters into the membrane, depolarization occurs, negativity decreases from minus 70 millivolt to minus 40 millivolt. Why this minus 40 millivolt is required? Because this is where the voltage gated calcium channels opens. Voltage gated calcium channel opens, calcium enters. Calcium is required for the exocytosis of acetylcholine, the primary neurotransmitter. Acetylcholine is primary neurotransmitter. Very simple. Okay, calcium causes acetylcholine release by exocytosis. This acetylcholine acts upon the receptors in the skeletal muscle to cause its function, that is contraction. So I have seen the excitation part. I have seen how different channels and different things aids in this process. We will see some facts about all this. So there is a voltage gated sodium channel blocker. Okay, this blocker is puffer fish toxin. This toxin is called tetrodotoxin. Okay, pufferfish toxin, tetrodotoxin. This is a voltage gated sodium channel blocker. This causes multiple paralysis and death. And death occurs due to multiple paralysis. Okay, first point. Second point is relating to the voltage gated calcium channels. So there are autoantibodies against voltage gated calcium channels. Okay, so there is no acetylcholine release, leading to paralysis. When muscle will not contract, it will ultimately lead to paralysis. This syndrome is called Lambert-Eaton myasthenia syndrome. Lambert-Eaton myasthenia syndrome, in which autoantibodies act against the voltage gated calcium channels. So there is no acetylcholine release by exocytosis, leading to paralysis. Third information we need to see is this release of acetylcholine. Okay, this really requires a protein called synaptobrevin. Okay, synaptobrevin, which is degraded by this toxin, very commonly known botulinum toxin. Called Botox. Botox. This causes a state of flaccid paralysis. Okay, this is acetylcholine release requires synaptobrevin. This is the single most protein here, which is degraded by our botulinum toxin. It causes state of flaccid paralysis. So these three important points we need to remember here. Okay, voltage-gated calcium channel blocker, copper phase toxin, tetrodotoxin, muscle paralysis, and Lambertian syndrome, Lambertian myasthenia syndrome, autoantibodies against calcium channel, voltage-gated calcium channels, no acetylcholine release, and no acetylcholine release requires synaptobrevin. Synaptobrevin is degraded by our botulinum toxin, causes flaccid paralysis. Okay, very important phase, need to remember by revising them. Okay, moving on, we have, so there is finally acetylcholine release. Okay, acetylcholine release. Now this acetylcholine has receptors here in the skeletal muscle. This is our skeletal muscle. This is called our nicotinic acetylcholine receptor. There are two types of acetylcholine receptors, nicotinic and muscarinic. So this is a nicotinic acetylcholine receptor. Acetylcholine release binds to it. Okay, sodium influx occurs, leading to depolarization. Okay, now this sodium influx causing depolarization produce two types of potentials. Okay, two types of potentials. We are going to see the two types. First is our end plate potential. Okay, this is due to smooth release of acetylcholine. Acetylcholine is gradually released as it should be due to smooth release of acetylcholine. It's a type of local potential. Okay, not action potential. End plate potential is not action potential, it's a local potential. Okay, now this can be summated. Okay, the end plate potentials generated, multiple end plate potential generations can be summated, after which it crosses the threshold. Okay, and leading to action potential firing. Initially it's not an action potential, it's only a local potential, but this can be summated, and after summation it crosses the threshold and finally leads to firing of action potential. This is end plate potential due to smooth release of acetylcholine. The other type of potential developed here is miniature end plate potentials or end plate potential, miniature end plate potential. Okay, this is due to spontaneous release of acetylcholine. Acetylcholine is released spontaneously in very quick successions. There is no smooth release, spontaneous release of acetylcholine. Okay, this is also a type of local potential. Okay, and none of these features of end plate potential are not present here. Okay, so this is all about the miniature end plate potential and end plate potential. These two types of potentials are developed due to sodium influx or depolarization in the skeletal muscle. Remember, end plate potential is not an action potential, but on summation there is an action potential firing. Okay, before seeing muscle contraction phase, we need to see an important discussion here. It is calcium channels in skeletal muscle. Calcium channels in skeletal muscle. Okay, calcium channels in skeletal muscle. So we'll take our time to draw a diagram first. Okay, so I'm drawing this diagram. Okay, now this we have drawn a diagram. Now let's level each parts. This first part we have drawn, this is our T tubules. Okay, this green part we have drawn here, this is our DHPR. What is transferred? Dihydropyridine receptor. Dihydropyridine receptor. Okay, what is this blue one? This blue one is a very important part. This is our RYR. What will is the RYR? We'll write in further. Okay, just remember this is our RYR, and this red color one, these are terminal systems. Terminal systems of sarcoplasmic reticulum. Terminal systems of sarcoplasmic reticulum. Okay. Now this RYR, which is a ryanodine receptor, we'll discuss further, and DHPR, dihydropyridine receptor, these are connected. Okay, connected by these strands here. So this is our entire structure of the calcium channel in the skeletal muscle. So brief structure. So what is this importance here? Let's see, when active potential reaches the muscle, okay, it propagates through muscle and reaches the T tubules. This is our T tubule arrangement, in which we have DHPR, RYR, and terminal system of the sarcoplasmic reticulum. The sarcoplasmic reticulum here contains calcium. We know this fact. This is our single most important ion. Okay, action potential reaches here and it activates this T tubule. Now there is a dynamic action between DHPR and RYR. Okay, now what action we are going to see here? DHPR, okay, is the key, acts as the key, and here are RYR, ryanodine. Excuse me, I'll write it again. Ryanodine receptor, or RYR is ryanodine receptor. Okay, this acts as a lock. So there is a lock and key mechanism between DHPR and RYR. Lock is our RYR, key is our DHPR. Okay, so these two have mechanical interactions. Mechanical interaction between DHPR and RYR. Okay, now when action potential reaches here, okay, we'll write in the steps. Action potential reaches and activates DHPR. Okay, Now, as the DHPR acts as the key, it's the key to opening the lock, that is RYR. Okay, this key, this DHPR, causes a conformational change. Activation of DHPR causes a conformational change in RYR. Okay, RYR. This opens the RYR, ryanodine receptor. This ryanodine receptor opening releases calcium from internal systems of sarcoplasmic reticulum. Here, this calcium releases. What is required? RYR. What is required for this opening of RYR lock? DHPR, which is the key. And what is the key? What activates DHPR? Action potential. Release of calcium, which will ultimately leads to contraction. Okay, very simple. Again, we are going to see action potential in the muscle reaches the T tubules. T tubules activates DHPR, which is our key to opening the lock, which is our RYR. Activation of DHPR causes conformational change in RYR. RYR opens and causes release of calcium from terminal systems of sarcoplasmic reticulum, which will ultimately lead to contraction. Okay, this was all about our calcium channels in our skeletal muscle. Okay, now we will see the ultimate phase after excitation or contraction. So muscle contraction. Okay, the steps. Okay, so we will draw diagrams and go one by one. Okay, so be with me. Very basic diagram I am drawing. Okay, so with this diagram we are going to level. Now this first step is at rest. Okay, is at rest. Now this is our, we'll start labeling. This is our actin protein, this is our myosin. Okay, this is our obviously myosin head, and we know what myosin head causes. Yes, ATPase activity. So this is our ATP. ATP comes in contact with the myosin head, which has ATPase activity. And very important, this angle between the myosin and myosin head is 45 degree. How this degree changes we'll see in further steps. Okay, now this is the entire thing at rest. Okay, what happens here? It is myosin head which has ATPase activity. This ATPase is activated when ATP comes to head of myosin. This is the exact statement given in the books. This ATPase is activated when ATP comes to head of myosin. The head of myosin having ATPase activity, ATP comes to here. Okay. Now as ATP reaches the ATPase, obviously there will be ATP hydrolysis. What will produce? ADP. From triphosphate to diphosphate, along with an inorganic phosphate will be released with help of ATPase. ATPase releases adenosine diphosphate along with an inorganic phosphate. This is our first step at rest. Okay. The second step we'll see again. Okay. So start drawing with me. This is our actin and this is our myosin. Okay, we'll draw the myosin head at once. Myosin head. So hereafter release, there is ADP and inorganic phosphate. This is ADP and this is our inorganic phosphate. Okay, myosin, this is our head, and this angle after hydrolysis of ATP becomes from 45 degree to 90 degree. Okay, this is our second step. This all happens in a second step. The angle changes from 45 degree to 90 degree, and ADP and inorganic phosphate attached to the myosin head. Till now there is no contact of myosin head and actin. Okay, how it comes we will see. After this second step, calcium comes. Calcium comes where? From sarcoplasmic reticulum. Sarcoplasmic reticulum, calcium comes. Okay, very important step. Okay, this is our actin, this is our myosin. Okay, so I'm drawing it all together. Okay, this myosin head finally gets attached to the actin. Okay, and here attached along with it are ADP and inorganic phosphate. Okay, this is our head, the myosin head attaches to actin. Okay, this step, the third step, is our cross bridge formation. This is a bridge formation between actin and myosin with the help of myosin head being attached to the actin, and the angle here remains 90 degree. Okay, this is our cross bridge formation. Now this head of myosin attaches to the here. What is this part is called? We know the active site of actin, which is we know from before discussion, which is covered by a tropomyosin, active site of actin. The head of myosin attaches to active site of actin. This is cross bridge formation or third step. Okay, after the cross bridge formation, ADP and inorganic phosphate are released. Okay, ADP and inorganic phosphates are released, and finally coming to the fourth step. It's not a final step, penultimate step. Okay, this is our actin. We'll magnify it through a thicker part. Okay, this is our myosin. Okay, and to which our myosin head is attached. Myosin head is attached. Okay, now magnifying this actin part, this is our actin. Okay, we see that this myosin head moves from this point to this point. Okay, moves from point 1 to point 2. So this is our head of myosin. So myosin head moves or slides from point 1 to point 2. Okay, so this actin part is called our sliding filament. Sliding filament, as our myosin head moves or slides from point 1 to point 2, is a magnifying part. So sliding filament part on which the head of the myosin slides. This event is called, given a specific name, remember, power stroke event. Here the angle changes from 90 degrees to 45 degrees. So this angle changes to 45 degrees, and this, all of this is called, yes, obviously, our sliding filament theory. sliding filament theory, which suggests that myosin, these are myosin thick filament, slides over actin is our thin filament. Thick filament slides over thin filament. This is our universal sliding filament theory, and this exact formation is called power stroke event. Remember, the angle changes from 90 degrees to 45 degrees. Thick filament myosin slides over thin filament actin. Okay, now after this step there occurs myosin head detachment. Myosin head detachment occurs. Okay, now after myosin head detachment occurs, new ATP molecule is required. New ATP molecule is required. Okay, and we are again back to the first step or fifth step. Here is your actin. Okay, here is our myosin. This is our myosin head. What is the angle? 45 degree. Myosin head of myosin. This is back to the normal, to the first step. Okay, so this was all about our contraction. Okay, this is all about our muscle contraction. I have tried to explain this in very simplified terms. It took a lot of time to design this actually, so please revise this part thoroughly to get the better understanding. Very simple concepts, nothing fancy. Okay, now we have a condition here. So after death, okay, after death there is no ATPs available. No ATPs available. But we have seen here, after the first sliding part, myosin head detachment, new ATP molecule is required, so that hydrolysis will occur and there will be cross bridge formation. Okay, so but no ATP is available after death. Okay, myosin head is still attached to actin. Myosin head is still attached to the actin. Okay, which is a very dangerous condition. It causes a state of sustained contraction. A state of sustained contraction. Myosin head is still attached to the actin. This is our actin. This is our myosin. This is our myosin head. It's still attached, so it will continue to slide back and forth, back and forth, back and forth. Slow, sustained contraction. Sustained contraction. This state is commonly called our what? Rigor mortis. Rigor mortis. Write this R once again. Rigor mortis. Okay, so after death no ATP is available here. Okay, no ATP is available. Myosin head is still attached to actin. Most important point here. So this called as a state of sustained contraction. It's called rigor mortis, which occurs after death. Okay, so this was our whole of contraction and our state of rigor mortis. Okay, now finally we will see the relaxation of skeletal muscle. Okay, relaxation of skeletal muscle. We know that the calcium ions coming out of the sarcoplasmic reticulum is the single most important ion in muscle contraction. Now for relaxation, okay, calcium ions needs to move back to sarcoplasmic reticulum. Sarcoplasmic reticulum. Okay, calcium ions needs to move back to sarcoplasmic reticulum. How these steps occurs, we'll see. Suppose this is a cell. Okay. Now we have our all important ryanodine receptor. Ryanodine receptor. Okay. This ryanodine receptor, this is our sarcoplasmic reticulum. Okay. This is the storage of calcium required for muscle contraction. Now this calcium move through this ryanodine receptor out. Release of calcium. Okay. Needed for contraction. Needed for contraction. Now this, after contraction, calcium ions needs to get back into the sarcoplasmic reticulum. For the transport back, we have a channel, and we have discussed this channel beforehand for relaxation, and this is our all important sarca pump. Sarcoplasmic reticulum calcium channel. All the calcium outside the sarcoplasmic reticulum will get back inside to the cell through our sarca pump. So two pumps in the sarcoplasmic reticulum are required for the mobilization of calcium. For a movement to out, ryanodine receptor; for contraction, after contraction, for relaxation, sarca to get back the calcium inside. Okay. Now in the sarcoplasmic reticulum, remember the terminal cisterns. I have also mentioned it before. Again mentioning, terminal cisterns is for storage of calcium. Storage of calcium. Okay, the terminal system. It's in this ryanodine receptor is attached to the terminal system of sarcoplasmic reticulum. Now, what happens in the defect? There is an ryanodine receptor overactivity. Ryanodine receptor overactivity. So there is enhanced release of calcium. Enhanced release of calcium. So there occurs more muscle contraction. More and more muscle contraction is occurs, which is not needed for our body. We don't need this muscle contraction, and this generates heat. This more muscle contraction generates heat. This produces a fatal condition called malignant hyperthermia. Malignant hyperthermia. It's a fatal condition which is due to, remember, ryanodine receptor overactivity. Ryanodine receptor overactivity causes malignant hyperthermia by generation of heat due to excess more of muscle contraction. It's a fatal condition. Okay. We have a ryanodine receptor blocker. Pharmacology, ryanodine receptor blocker. And what is that ryanodine receptor blocker called? Dantrolene sodium. Dantrolene sodium is a ryanodine receptor blocker. This is in treatment of malignant hyperthermia. So this was all about our discussion of the properties of skeletal muscle. Okay, we've seen the electrical events, the action potential, the contraction phase, and before that we have seen a sarcomere, changes in the sarcomere, the proteins in the sarcomere, contractile proteins, regulatory proteins, supporting proteins, and relaxing proteins. Okay, very important topic in physiology. Forms the basis of many more further chapters and subjects in further years of MBBS. So keep revising them. Thanks guys. Thanks for watching. I will catch you in the next class. Good luck.